Well, again, thank you very much for the kind introduction, Sarah, and also for the invitation and the opportunity to present my work here today. I would like to start by giving you a short introduction into chromatin. And as you know, our DNA is packaged tightly into chromosomes and stored in a tiny nucleus full of proteins and other molecules. And to achieve this tight packaging, our DNA is wrapped around so-called nucleosomes, which consist of different histone proteins. Those histone proteins can be enzymatically modified with a variety of chemical marks on their flexible N-terminal tails, but also on their globular part of the histone proteins. And in some cases, also some modifications can influence genome organization by forming also spatially restricted compartments. And so this is the case for HRK9 trimethylation and HRK27 trimethylation, which are both repressive histone marks and the hallmarks of constitutive and also facultative heterochromatin. And so from research over the last years, we know that in many cases, these modifications regulate genome function by recruiting specialized reader proteins to chromatin. And to understand how these chemical modifications drive genome organization and also regulate genome function, we think it's crucial to address two current challenges. First, to identify which proteins associate with the genome based on the local chromatin modifications. And second, to dissect how reader proteins bind to a chromatinized genome. And so in this study, we present new methods to address both challenges. We developed so-called engineered chromatin readers, which are selective towards individual and also combinatorial chromatin modifications. We now use these engineered chromatin readers for several biological applications, such as the dynamics of histone modifications in living cells, their genome-wide mapping, but also, for example, to dissect the histone mark crosstalk that exists in mammalian cells. And in the last example, I would like to show you how we can couple selective chromatin readout with proximity biotinylation to identify local proteome composition at individual and also combinatorial chromatin marks. And so to build engineered chromatin readers, we made use of naturally occurring protein domains that are selective towards histone modifications and also DNA methylation. And so we first started by stably expressing well-characterized reader domains in mouse embryonic stem cells, for example, the chromodomain of CBX7, which is selective towards K27 trimethylation. And so we express this either as a single or a double domain GFP fusion. And we first measured their subnuclear localization by light imaging and found that multivalent interactions are required to yield selective histone mark readers, as shown in these two examples. So when for example, we express two chromodomains of CBX7 fused to GFP, this resulted in a very distinct nuclear localization pattern that resembles very much to what people observed with immunofluorescence and specific antibodies towards K27 trimethylation. And in comparison, when we only express one chromodomain fused to GFP, this results in a very diffuse nuclear localization that is similar to what we observe with a nuclear GFP control. And so again, multivalent interactions are required to yield selective histone mark readers, and this is an observation that has also been made by other labs in the field. We could also further demonstrate the selectivity of our engineered chromatin readers towards their target chromatin modifications by ChIP-seq. So here I'm showing you snapshots of the genome, showing all the different readers that we generated and also their corresponding target modifications. Here in the top two tracks, you can see, for example, the binding of our K4 trimethylation reader and the corresponding target modification, K4 trimethylation. And as you can see, there's a very good overlap between the two. And this we can also observe more globally. So this, with this and others experiments, we could show that ECRs are indeed highly specific towards their target chromatin modifications. And so now here I would like to show you an example of how we can use ECRs fused to GFP to study, for example, the dynamics of histone modifications in living cells. And so here I'm showing you two videos. So in the left, you see cells stably expressing the our reader that is selective towards K27 trimethylation. And on the right side, you see the nuclear GFP control. And something that really catched our attention is when you follow, for example, this cell marked in yellow. When the cells start entering mitosis, you see an accumulation of and the appearance of these dots towards the periphery. And this we observe for every cell that enters mitosis. This you can probably appreciate more in this close-up. And so we're very curious to figure out what the structures are and whether they have any biological function. But I think it nicely, this example shows how the potential of using ECRs to study the dynamics of histone modifications in living cells. Now, coming back to what I told you in the beginning, so one of the challenges that we want to address is to identify which proteins associate with the genome based on their local chromatin modifications. And so for this, we exploited the binding specificity of ECRs to establish what we call Chrom ID. And so in Chrom ID, we express ECRs now fused to a promiscuous biotin ligase, and which can be activated upon the addition of biotin to the medium. And so this results in the biotinylation of all proteins in a certain radius and a certain proximity that are proximal to the target chromatin modification. And so this strategy allows us to isolate all biotinylated proteins and detect them via mass spectrometry. And also importantly, since this labeling occurs in living cells, this method also allows us to preserve many relevant molecular interactions. And so we first started to optimize the conditions for Chrom ID by studying the proteins associated to H3K9 trimethylation, since this mark has been very well studied in the last couple of years in mammalian cells, so we knew what kind of proteins we could expect from these experiments. So here I'm showing you a volcano plot summarizing 4 independent Chrom ID experiments using our K9 trimethylation reader. And as a control, we use a reader-free nuclear biotin ligase. And so all the proteins named here are proteins that we identify significantly enriched using our K9 trimethylation reader. And we took all these proteins, performed GO term analysis, and the top GO terms that we can identify are all linked to heterochromatin, which again validates the sensitivity of our method. But we also took one zinc finger protein that has not been characterized so far in mammalian cells, at least not to our knowledge. This is selenF280D. So this is, as I said, a zinc finger protein. So I tagged this protein endogenously and performed ChIP-seq analysis. And as you can see, it gives beautiful binding peaks. We then looked more closely so that we can collapse all the binding peaks and look what kind of histone modifications are enriched around those binding sites. And as you can see here, we see a very strong enrichment for H3K9 trimethylation at all these binding sites. So again, validating the sensitivity of our method. We were also curious to establish or engineer combinatorial ECRs that don't exist like this in nature. And one motivation to do this is because we also would like to target combinatorial chromatin modifications. And one of those modifications or combinatorial chromatin modifications that are very interesting and important to study is bivalent domains. So bivalent domains mark essential developmental promoters, and addressing their genomic distribution and also protein composition has been very difficult in the past because of the lack of tools that enables the simultaneous detection of K4 trimethylation and K27 trimethylation on the same nucleosome. And so to overcome this limitation, we decided to take the chromodomains of 2 well-characterized proteins, namely the Drosophila polycomb protein or the mammalian CBX7 protein. So both chromodomains are selective towards K27, and this we couple to a PhD domain of TAF3, which is selective towards K4 trimethylation. And this resulted in a selective bivalent reader. So here I'm showing you again a snapshot of the genome summarizing what we observed in independent ChIP-seq experiments. So here on the top, I'm showing you antibody chips to K4 trimethylation, K27 trimethylation, and here the binding of our K4 trimethylation reader, the K27 trimethylation reader, and 2 bivalent readers. And as you can see, the bivalent readers nicely localize at regions in the genome that are bivalently modified with K4 trimethylation and K27 trimethylation, whereas other regions in the genome that are only modified or only marked with K4 trimethylation are not bound by these 2 readers. And so now, obviously, we took all the readers that we generated and performed Chroma ID experiments, and this resulted in high confidence proteomes of all the key chromatin states that I mentioned to you, or that I showed you. Key chromatin states in mouse embryonic stem cells. And some of the highlights from this experiment, so we were able to discover novel proteins and also measure their binding preference. And one of these examples I showed you before, the 7F28D. We could also identify a previously unknown crosstalk between the proteomes of K27 trimethylation and K9 trimethylation. So there are a bunch of proteins that are shared between these 2 chromatin states. And lastly, we could also uncover the proximal interactome at bivalent promoters. And so with this, I hope I can convince you that so first of all, we developed engineered chromatin readers that are selective towards individual and also combinatorial chromatin marks, and we use those readers for a variety of applications in living cells. And now, as an outlook, I would like to tell you how we use similar strategies to identify the proximal interactome of transcription factors. And so we chose CTCF as a starting point. CTCF is the prevalent insulator protein in mammalian cells. And so we took the DNA binding domain of CTCF and coupled this to turbo ID, which is another promiscuous biotin ligase. And this we express in the presence of the endogenous CTCF protein. So now, upon the addition of biotin and labeling for 12 hours, we can capture what we think are cohesin-dependent and also cohesin-independent interactions. And so this is how the data looks like. Here again, this volcano plot summarizes 4 independent experiments, and on this side you see the proteins that we significantly enriched with the DNA binding domain of CTCF. And as you can appreciate, we detect or we can identify components of the cohesin complex, but also other proteins that are now have been recently identified to be competitors of CTCF. So these proteins, ADMP and WIS, compete with CTCF for the same binding sites. And so we're very excited to use similar approaches to identify more proximal interactomes of transcription factors, and also in this case, in the case of the CTCF proteome, we will dig further and analyze other interactions. So with this, I would like to acknowledge all the people involved, Tuncay for all his support in the last couple of years. And I would also highlight here Ramon, which was the one who developed the Chroma ID protocol in the lab. And I would like to thank you for your attention, and I would be happy to take any questions.
Two very small questions. Yes. So one very small question is about the enrichments in the area of H3K9 trimethylation. Did I not notice HP1 there, or?
So the HP1 is there. So we can identify CBX1 and also CBX3. CBX3 does not fall above the threshold, but it's also there somewhere in the cloud. The problem with CBX1 is, so we are using the chroma domain of CBX1 for our reader. So we cannot really differentiate between the reader or the endogenous HP1 protein. But what I can tell you is that we also, so if we look at proteospecific peptides from endogenous HP1, we also detect those in our pull downs.
Yes, hi Rodrigo, wonderful work.
Thank you,
I was
Maria.
wondering, does expression of these ECRs have any kind of effects on cells? Does it cause, function phenotypes because you're competing with the
Yes.
endogenous binders?
Yes, you're completely right, and this was indeed something we were asked. I prepared here a slide. So these cells essentially they can differentiate to terminal neurons. So all of the cells that essentially express these readers are able to differentiate, and at least we don't observe any severe abnormalities with these cells. But this might obviously depend on the reader that you take. For example, and this might also depend on the amount of targeted modifications. So in the case of K9 trimethylation and K27 trimethylation, these are very abundant marks. And so there we don't observe any, or we are, let's say the reader can compete also with endogenous proteins for many binding sites. But I think we now started looking more closely into our K4 trimethylation reader there. I think we probably need to lower the expression levels to avoid any competition, as you mentioned. But essentially, all these cells that overexpress these readers are morphologically fine, and they can also differentiate towards terminal neurons.
Thank you.
Yes, hello, Rodrigo. Very nice presentation. I was just wondering if when you look at bivalent mark, can you basically see if the two marks are on the same tails? Eventually, if there is, if you are looking at two marks that may be on the same tails, or it's, or you cannot distinguish between these two properties.
No, yeah, you're right. So we can't really distinguish. So we don't have this resolution, and it's our assumption essentially that we are targeting the same nucleosome, but we can't really exclude the possibility that we might probably bridge two neighboring nucleosomes. But I think there is good evidence to think that this bivalence or bivalency actually that it exists on the same nucleosome. So there is beautiful work from Philip from Danny Reinberg's lab essentially, and also from Efrat Sharma using single molecule studies. So I think it's fair. It's probably safe to say that these modifications exist on the same nucleosome, but we can't really tell at least with our tools that we are capturing mononucleosomes.
Okay, thank you.
Hey, Rodrigo, very nice talk. Thank you very much.
Thanks, Manuel.
And I want to ask you, did you check also some in vivo imaging about the bivalent distribution into the cells?
Yes, we did. Is actually so it shows a very diffuse nuclear localization pattern, but if we do see signal on the condensed chromosomes, basically it stays bound to chromosomes.
But it's not is having a special distribution towards the nuclear.
Unfortunately not. We don't, at least not with the conventional imaging that we're doing, so with confocal microscopy. But we are actually very excited about now the possibility of using more super resolved methods to and address the same question, actually, to maybe start seeing some patterns there or structures that we are not able to see using conventional confocal microscopy.
So I have a small question myself while we are waiting for the others. I was very interested about ADMP CTCF link. So if you say ADMP and CTCF compete, then why would we observe actually ADMP enriched together with CTCF? Wouldn't they be like expected to be separated?
Well, to be honest, I'm also surprised about this. Seeing competitors of CTCF. I cannot give you a very good answer. but I think I think we're really entering in a competition, at least with this construct CTCF or the DNA binding domain fused to the turbo ID construct. This somehow competes within the endogenous CTCF, and maybe this competitions or on and off binding are so fast that allows to capture this kind of interactions. But I cannot, to be honest, I cannot give you a good explanation for why we do observe and capture ADMP.
From Dominique, who is asking, hi Rodrigo, thanks for the great presentation. Can you speculate why ECRs with only a single binding site have low specificity?
Yeah. Hi, Dominic. So this is a great question again, and I think this has to do with the symmetry of nucleosomes. So I think if you imagine nucleosomes being modified on both tails, this might be the case why our dual readers are actually capturing these interactions and are being more or are able to capture this kind of modifications. The single domains might just simply not be sufficient, or might not have enough affinity to recognize modified nucleosomes. But this was actually very striking. Essentially, for all the histone mark readers that we generated, all of them only work when we couple two domains with each other.